study sponges because these are the most ancient metazoans we know, the first animals, so we want to look into the origins of animals by studying sponges. Sponges are very simple animals, and sometimes they don't show the same structures as we see in other animals. But not having the structure doesn't mean that the sponges do not have the genes. So by looking at these genes in sponges, we can understand the complexity of these structures in other animals. So we have analyzed the transcriptome of 8 different species of sponges because it's a little less complicated to analyze than the genome. And it also gives us advantages in knowing what are the genes that are expressed in a given moment for any species. So the genome of a species contains all the genetic information of a given species, but not all this information is expressed, and we are only interested in this expressed information, and that is actually the transcriptome. So actually we have found many different genes that were not expected in sponges. Concretely, 19 genes that were involved in neurotransmission, coagulation, or rapid contraction responses. So for instance, we have found glutamate receptor genes that are usually involved in contractions in other animals, and in sponges they are involved in the contraction response to the clogging of sediments in the canals of the sponge, so the sponge has to get rid of this sediment by contracting and expanding. So this discovery is indicating that sponges can respond rapidly to different stimuli of their environment, and until now this is something we didn't think sponges could do. Actually, what we found is that sponges are genetically more complex than we expected. From now on, our challenge is knowing what are the processes these genes are involved in, and what are the structures they form in sponges, because right now we only know those things about a couple of them. We can understand the rest of the processes in animals by studying sponges because these are the processes most simplified. So by studying sponges we can understand more complex processes. Like for instance the epithelia. If we look at the epithelium in sponges, it's a very simple thing, but we can understand this and then understand how the rest of the animals build upon these different simplifications. So our priority right now is understanding where these genes are expressed in the body of a sponge, because by this we want to understand how these genes are involved in more complex structures in other animals.